Good day, ladies and gentlemen. I am Genevieve B. Portarcas, and I will be presenting to you the process of DNA replication. DNA replication is the process of producing two identical copies of DNA from two old DNA strands. Double-stranded DNA is a polymer of two strands of nucleotide held together by hydrogen bond to form a double helix. The building block of DNA is called nucleotide. It consists of three important elements: a deoxyribose sugar, phosphate group, and a nitrogenous base. The backbone of DNA consists of alternating sugar and phosphate groups that are held together by phosphodiester bond. The nitrogenous bases include cytosine, thymine, adenine, and guanine. Cytosine paired only with guanine and forms three hydrogen bonds, while thymine forms two hydrogen bonds with adenine. Reason why cytosine and guanine are more stable in compared to thymine and adenine. One strand of the DNA is oriented to 5' to 3' direction, and the other strand is oriented to 3' to 5' direction, since DNA strands are anti-parallel to each other. In replication, each strand of the DNA is used as a template in creating new identical copies of the DNA. Replication starts as the double-stranded DNA being separated from each other, having the two single-stranded DNA called the parent strand is used to make a new complementary strand. DNA replication occurs in 5' to 3' direction, adding new nucleotide to the 3' end of the growing strand. DNA replication begins at a specific site or location called the origin of replication. To understand how complex eukaryotic organisms replicate their DNA, scientists first studied prokaryotic cell replication using E. coli. There are various enzymes participating in the process. The process begins when an enzyme called helicase binds to the origin of replication and starts to unwind the DNA, forming a replication fork. A protein called single-stranded binding protein prevents the two strands from annealing or forming hydrogen bonds. Unwinding the two strands causes torsional strain in the helix. So, to remove the strain, DNA gyrase, a subtype of topoisomerase II, will uncoil or untwist the DNA ahead the replication fork and prevents it from supercoiling. Having the two strands separated, we can now begin the replication process of each strand. An enzyme called DNA polymerase III catalyzes the addition of new nucleotides. This activity is called polymerization. Other proteins such as beta clamps and a clamp loader help hold the DNA polymerase into place. To start the polymerization process, an enzyme called primase will anneal a short sequence of RNA called RNA primer that is usually 5 to 10 nucleotides and complementary to the nucleotides in the parental DNA. This mark for DNA polymerase III to start the polymerization process from 5' to 3' direction. RNA primer is needed because DNA polymerase cannot begin to add nucleotides without it as a starting point. In this phase, DNA polymerase III binds to the primer and begins to add new complementary DNA nucleotides, creating a new copy of DNA strand. The replication of the two strands occur at the same time. One is in a continuous synthesis while the other is discontinuous. Since DNA polymerase replicates solidly in 5' to 3' direction, continuous synthesis occurs on the 3' to 5' oriented parent strand is referred to as the lagging strand. New nucleotides are added in the 3' end of the growing strand. On the leading strand, the replication is continuous. Primase must add new primers every after 100 to 200 nucleotides, creating chunks called Okazaki fragment. This means that we need one primer in every Okazaki fragment, which also means that not just one primer is needed in the lagging strand. An enzyme called DNA polymerase 1 removes RNA primers from the DNA strand and replaces it with DNA nucleotides to make sure it is DNA all the way through. Another enzyme called ligase filled the gap between nucleotides to make sure everything is connected. Two new DNA strands are produced at the end of the process which are identical to the parental strands. Note that DNA polymerase 3 moves only in one direction and it is always add nucleotides from 5' to 3' direction, reading from 3' to 5' end of the parental strand. That's why the direction of replication of both strands are opposite, since the strands are anti-parallel.